Good afternoon, children, and welcome back to biology class. Last week you saw how a recombinant DNA is made, okay, and how this recombinant DNA how the bacterial cells are made competent to take up this recombinant DNA, right? Now, when inside this bacterial E. coli or bacterial cell, this will start multiplying. This plasmid along with the recombinant DNA will multiply inside the bacterial cell and make copies depending upon the number of copies. Okay, now if you allow them to grow and if you use all the how many pieces of recombinant DNA if you have made by PCR, all you incorporate onto plasmid and insert inside the bacterial cell, and even if you allow it to grow in bacterial in lab conditions, how much of the the products you will get? You get only in few milliliters. That much amount only you will get out of these bacterial cells. In a very less quantity wise, it is only less. Okay. Now let us see. The ultimate aim of recombinant DNA technology is to produce a desirable protein. Whatever recombinant DNA we have incorporated, that is what they are talking about. Desirable protein. If a protein encoding foreign gene is expressed in a heterologous host, it is called as recombinant protein. Okay, recombinant DNA or protein. Now, instead of using a bacterial cell, we can use a bioreactors. A bioreactor is a large vessel where you can get large quantities, nearly 100 to 1000 liters of the culture of the product, whatever product we are intending, that product we can get. 100 to 1000 liters. Okay, whereas the bacterial, if you allow them to grow in a lab condition, we get only few milligrams of this same product, right? So to get continuous and large quantities of this recombinant protein, we use what is called as a bioreactor. Okay, so cells with foreign genes can be grown in laboratory. The cultures are used to extract desired protein and purify it by using separation techniques. The cells can also be multiplied in a continuous culture system. Here, the used medium is drained from one side. Used medium means after this recombinant DNA. See, this is the bioreactor. Okay, where you can supply through one side you can supply the raw materials. Okay, and on another side you can derive the product. Okay, you can control the oxygen bubbled into that. You can control the temperature of the whole vessel. Okay, everything can be controlled. Okay, right. So a continuous culture system. Here the used medium is drained out from one side while fresh medium is added from the other. It maintains the cells more physiologically active and so produce a large biomass. It yields more desired protein. Okay, children? Right. Now let us see what are these bioreactors. See, these are the vessels in which enzymes, products, etc. are made using microbial, plant, animal or human cells. These are large vessels, children, where raw materials are converted into useful products or enzymes or whatever it is. Bioreactors are used to produce large quantities of products. They can process up to 100 to 1000 liters of culture. A bioreactor provides optimal growth conditions, which is temperature, pH, substrate concentration, salts, vitamins, oxygen, to get desired product. Everything can be adjusted. All these levels can be adjusted in a bioreactor. Okay. Now, the most commonly used bioreactors are, children, two types are there. They usually ones used are the simple stirred tank bioreactor and a stirred and sparged type. Okay. Now, most commonly used bioreactors are the stirred tank reactor. It is usually cylindrical or with a curved base to facilitate the mixing of the reactor contents. The stirrer facilitates. Why it is stirred? Always continuously it is kept stirred. Stirred means, continuously agitated, shaking. Okay. Now, the stirrer facilitates even mixing of oxygen as well as oxygen availability. Alternatively, air can be bubbled through the reactor. Okay. The other one is the sparged one. Okay. Whatever is the case, what are the other conditions of these bioreactors? What are the other facilities in the bioreactor? It has an agitator system which is kept continuously agitated, an oxygen delivery system, a foam control system, a temperature control system, pH control system, and a sampling port. Sampling port is for collecting our desired product. ഒരു ഭാഗത്ത് നിന്ന് നമുക്ക് product we can collect it. Okay, that is the sampling port. Clear, children? Now, after getting the products are not in its pure form. What do you mean by the products now? What products do you expect from these bacterial cell or animal cell or whatever it is? It is usually you might have studied the next chapter, that is applications of biotechnology, isn't it? Right, that's it. So we make insulin out of the bacterial cell. We can make proteins. We can make enzymes. We can make vaccines, isn't it? So these are the different antibiotics. We can make antibiotics. So these are the different proteins or enzymes made or derived out of bioreactors. Okay. Now, when you get these products, it is not in its pure form. It is mixed with many other components inside the bioreactors. So we have to, what's the next step is, we cannot directly use the product. It has to undergo certain purification process, which are collectively called as downstream process. What is that? Downstreaming process. This is a series of processes such as separation and purification. Sorry, children, for the disturbance. Okay. So what is the downstreaming process? Downstreaming process includes two main techniques, which is separation and purification of the products. What are the two techniques or two processes included in downstreaming process, children? Separation and purification. Okay, the product is, if it is a vaccine, suppose if it's a vaccine is the product you get, right? Vaccines cannot be directly used for human beings. It has to be undergone clinical trials. It has to be tested in the case of for monkeys or rabbits, and then only if it is success only it will be used for giving it for human beings, right? Okay, if it is some other products, or if it has to be preserved for many days or many months together, it should be added with certain preservatives, right? Chaladokke namukku directly we can use it as, after purification, after downstreaming process, we may use it directly. So according to the products, different downstreaming process need to be done. Anyway, two techniques are involved. Which are the two techniques, children? Separation technique and purification technique. Okay.